modes of genetic exchange. Very briefly, I went through the three modes, which are conjugation, transduction, transformation, and briefly provided a simple diagram for transduction, transformation, also for conjugation, but then I went into more detail into conjugation, the proteins that are involved, and you have to know that. And what are plasmids? How are plasmids involved in this process? And how the plasmid moves from one cell to another. In other words, what are the proteins that are involved in that process? Then I ended up by talking about the integration of the actor, one of the first plasmid that was identified in bacteria, to create HFR strains, and how these strains are very useful in mapping. Okay. I'm not going to be talking about mapping. I want to talk about another bacterium and a plasmid that it has and how it modifies plants by transferring DNA via conjugation type mechanism. So I am in the same topic, conjugation type of transfer, but in a different organism. I will go back to mapping later on, not today. Okay, so then, oops, why is this going like this? Okay, hold it a second, sorry about that. Oh, I see. Okay, go back to the. Okay, today I'm going to be talking about Agrobacterium tumefaciens and DNA transfer across domains. So from bacteria to plants. Remember that there are three domains. There is bacteria, archaea, and eukarya. Plants are in the eukaryotic group. Bacteria belong to another domain. And in this case, Agrobacterium transfers DNA to plants and modifies them. And that is the basis of genetic engineering in plants. Of course, there is a new technology now that people are using, but people still use this. Okay, so what is Agrobacterium? It is a soil bacterium and is related to Rhizobium, the one that is involved in nitrogen fixation, and Rickettsia, which is a pathogenic bacterium. Okay, it is a gram-negative. It causes a disease called crown gall disease. Okay? This has been known for a long, long time. And this disease is characterized by tumors. So these are outgrowths on the plant, and tumors are really unusual in plants. Okay, so there are only a few organisms that can cause tumors in plants, and Agrobacterium tumefaciens is one of the few. These are the pathogenic agrobacteria contain a 200 kb plasmid, which is called the TI plasmid, and we are going to be talking a lot about this plasmid. And Agrobacterium, very importantly, okay, let me get this, the arrow, infects only wounded plants. And we are going to see why this is very important in the process. It's really a very interesting story, and one that in general people don't hear about, but it's a fascinating story. So Agrobacterium will transfer part of this plasmid, not the entire plasmid, to plant cells in a process that is similar to conjugation. And the transferred DNA will integrate stably in the plant chromosomes, so it integrates in the plant chromosome. And this DNA that integrates contains information for production of plant growth regulators. These are hormones. And also for production of special amino acids. Okay, these are new amino acids. You never heard of these amino acids before. And these amino acids are used by the agrobacteria as a carbon and nitrogen source. So the bacterium induces the plant by transferring this DNA to produce these amino acids. But only agrobacteria that contain the TI plasmid can utilize these amino acids as carbon and nitrogen source because they have the genes in this plasmid that can break down these amino acids to be used for this purpose. No other bacteria will have it. Okay? So these amino acids that are also produced are also involved in transferring the TI plasmid to other bacteria. Okay, so the bacteria are very smart because in nature, Agrobacterium tumefaciens, not all of the Agrobacterium tumefaciens population contains the TI plasmid. Okay, so, but they need to, once they have contacted a wounded plant, it is very important that the population acquires the plasmid. And there is a reason why not all bacteria all agrobacteria, I should be very specific, agrobacteria contain this plasmid, and I will talk about that in a moment. And it is because bacteria, the Agrobacterium tumefaciens containing this plasmid, the TI plasmid, have a selective disadvantage in the presence of other bacteria in the soil. Remember, Agrobacterium is a soil bacterium, normally lives there very happily, doesn't have to be in the plant. But if it has the TI plasmid, then other bacteria produce a toxin that will kill Agrobacterium. So if you have all the population of Agrobacterium tumefaciens with the plasmid, then they are all going to be killed. Okay, so you only have to have a few of them, and then once the conditions are appropriate for them to transfer the plasmid, that is when they infect a plant, then they can transfer like crazy the plasmid to other bacteria because they are using these amino acids that they induce the plant to produce to transfer the plasmid via conjugation. Okay, so the first, yes?
So the TI plasmid, the bacteria having the TI plasmid will kill other bacteria or the other?
No, they get killed by other bacteria that are not agrobacterium. Okay, so agrobacterium, I should have had a simple diagram, I see. So Agrobacterium tumefaciens, when it is in the soil, there are other soil. Remember, there is a vast population of bacteria that are also inhabiting the soil. Some of these bacteria, I don't remember the species, produce a toxin, agrocin, and this toxin kills bacteria that have the TI plasmid. So Agrobacterium tumefaciens without the plasmid survives. Agrobacterium tumefaciens with TI plasmid is killed by this toxin. So if you want to maintain this species, only a few should have the TI plasmid, unless they are infecting the plant. Okay, so Agrobacterium tumefaciens have made possible modern molecular genetics of plants and the modification of plant genomes to produce, and I will say in quotation mark, agricultural useful plants. This has been a very controversial topic, and I can address it at the end of this, when I finish lecture on Agrobacterium. Okay, so here are some tumors that are induced by Agrobacterium tumefaciens. This on the stem. So you see they can get pretty big. This is in a tree. And these are stems of plants. And here to really show you how big they get, this is a person next to a tumor on the base of the stem of a tree. Okay, so they can get really huge. And this is all due to Agrobacterium tumefaciens. So what is this famous plasmid, the TI plasmid that this bacterium contains? Okay, so this in a cartoon form, this plasmid has several regions. Of course, it has an origin of replication or EB. It also has an origin of transfer, which I'm not showing here, which is involving conjugal transfer. Conjugal transfer refers to conjugation. It has a region that is called the Vir regulon, which I'll describe later. It has the trigenes, so these are important for conjugation, but not to transfer into the plant, okay? This is for conjugation between different agrobacterium tumefaciens cells, okay, from one to the other, not to the plant. It has genes for opin catabolism. Opins are these different and new amino acids that are induced by agrobacterium. So catabolism meaning the breaking down of these amino acids to be used as carbon and nitrogen source. And then here you have the tDNA, the transfer DNA. Of this plasmid, the only thing that gets transferred to the plant is this block in red. Okay? So that's where we are going to analyze. So what does this tDNA have? So tDNA for transfer DNA. And this is what I'm going to be describing now, just this. Okay, so it has a block, several genes here, a block of genes. And I'm going to tell you what they are. So here, the tDNA has 25 base pair repeats at both ends. One is called the TR and TL or LB for left border, right border. Either term is fine, okay? Whatever you want to use. But these are 25 base pair repeats, which are very important in the transfer process. Okay, so inside this, these repeats, you find this set of genes. There are multiple genes, and I'm going to tell you what they are. So IAA stands for auxin biosynthesis. IPT stands for cytokinin biosynthesis. Please pay attention. Cytokinin is different from cytokine, which is present in humans and other mammals, and this is involved in the inflammatory response. The cytokinin has nothing to do with inflammation in plants, okay? So make sure that you understand the difference and spell it correctly. And opin is opin biosynthesis. Opin are special amino acids. And you had in your exam a question about nopalin. Nopalin is produced by one of these TI plasmids, and it's of the group of the opins, okay? So it is an unusual amino acid. Okay, this region is what called for synthesis of these amino acids, and in the TI plasmid, you have catabolism. This is how you break that codes and makes these amino acids, codes for the enzymes that make these amino acids. Here you have the region that breaks them down. Okay, those are two different regions and are going to reside in different places. Okay, so auxin, what is auxin? Is a plant growth regulator. And cytokinin is another plant growth regulator. So these are really important in determining whether, the morphology and whether plants form shoots, roots, whatever. You can manipulate this in the lab. So the ratio of auxin to cytokinin is very important for shoot and root development and normal plant growth. Okay, so here shows that how this ratio can manipulate the ratio. You can take a plant tissue. Depending on the ratio, you can just make this grow into a mass of undifferentiated cells. They are meristematic cells. These are undifferentiated cells. Or with another ratio, you can make roots only. So you can grow only roots. I know I did it in the lab when I was a student. So, and then under other conditions, you can just make shoots with no roots. Obviously, this medium will have the appropriate nutrients so you can grow this. And then in other cases, you have no growth.